Great. Well, thanks for having me here.
Yeah, sure.
Yeah, normally I do these remote, right from home, but we
Yeah, it's
both.
cool that we're, geographically co-located that we can do it in person.
Yeah. Oh, where am I right now?
What do you mean?
What is this place? What do you
This
do?
is OptiSpan offices, and then right next door is Aura Biomedical. So they're a company we spun out of my lab when I was at UW. They do high throughput longevity drug discovery using robots and worms. We can go take a look over there when
Okay.
we're done if you
Yeah, that would
want
be
to.
great. That'd be great.
Yeah.
Do you just want to start off by telling everyone a little bit about who you are, what your scientific background is?
Sure. So my name is Matt Kaeberlein. I am the CEO of OptiSpan. I guess I would describe OptiSpan as a healthcare technology company. So our mission is really to create tools, technologies, protocols to enable science-based, proactive, preventative healthcare for as many people as possible. So we are very much of the belief that the current healthcare system is not as effective as it should be, to put it mildly, and that really it's centered around what I would call reactive disease care. Typically, we wait until people are sick before we try to do anything about it, and we try to treat their disease. We really want to help enable more proactive, preventative healthcare by both detecting disease early, but more importantly, by empowering people to keep themselves healthy. And so I spent, most of my career until about a year ago in academia studying the biology of aging. I think that the biology of aging is certainly the most important risk factor for most functional declines and diseases that people experience. So I think one aspect of this transition to what I call 21st century medicine, what Peter Attia would call medicine 3.0. I think targeting the biology of aging is an important part of that, but it's only part of it. There's also, a huge component that involves screening for problems before they happen, and taking steps to ensure that we've sort of optimized our physiology as much as possible to maintain health. So I think you need all of those pieces, and that's really what we're all about here. So like I said, my background really is I'm trained as a research scientist, and my entire career in research was spent trying to understand the mechanisms of biological aging, and why is it that all animals appear to undergo physiological decline as we get older, and what are the mechanisms and what aspects of those mechanisms are shared across the animal kingdom, with the expectation that the mechanisms of aging that are similar across the animal kingdom will be relevant in people.
hmm Yeah, one of the things that's super interesting is if you take a comparative approach to aging and you look across many different species, you think about this in evolutionary terms. All animals age. There's important differences between certain species, but something that's very interesting that I've talked with others about is there is this very lawful relationship between how long an animal lives, how big that animal is, and its metabolic rate.
Yeah.
Right. So small. This is why we talk about dog years.
Yeah.
Our dogs age faster than us.
Right.
Small animals tend to age more quickly. They have shorter lifespans. How do you think about that relationship? Why does that relationship exist, and what does that start to point us to?
Yeah. So great question. It's actually more complicated than that, because when you look across species, what you said is true. Smaller species tend to age more rapidly, have a higher metabolic rate than larger species. When you look within species, it's actually flipped. Smaller individuals tend to age more slowly. And we understand, I think, the mechanisms of within species variation based on body size pretty well. So we can get into that if you want to. I think we don't really understand the mechanisms for a cross species relationship between body size and longevity. So certainly that sort of metabolic rate of living hypothesis is one hypothesis that's been around for a long time. Essentially, the idea there is that the faster you're burning metabolism, the more rapidly you accumulate damage associated with metabolism, the more rapidly you're going to age. That's almost certainly part of the story. I don't think it's the entire story. We know of species that kind of seem to break that correlation a little bit.
Yeah.
So there's more than that going on. I think And I think this is really one of the mysteries in the field right now, because again, if you look at the within species variation, I mean, dogs are almost an outlier where within the same species you have about a twofold variation in lifespan based on body size. In most species, it's much less than that. But if you look across species, we're talking many orders of magnitude variation from very small animals to very large animals. So that variation in rate of aging and longevity across species is much larger, and we really don't understand the mechanisms. And the hope would be if we could understand the mechanisms, that might offer opportunities for therapeutic approaches that are much larger effect size than what we have available today. I think one thing that's worth saying, though, is from an evolutionary perspective, even though this isn't satisfying from a sort of molecular mechanism perspective, from an evolutionary perspective, it kind of makes sense because larger animals tend to reach sexual maturation later. So you have to evolve mechanisms of slower aging in order to be successful from that sort of evolutionary perspective. If you're not going to reach sexual maturation for months or years, in the case of humans, for example. The other thing that's worth appreciating is larger animals have more cells, so they have to evolve more robust anti-cancer mechanisms.
Because they have so many more opportunities for cancer
Exactly,
to arise.
right? Just from a stochastic perspective of a cancer-causing mutation arising, you have a lot more opportunity for that to happen if you have, 100, 1,000, 1,000,000 times more cells.
So if a whale had the cancer detecting and cleanup capabilities of a mouse, it wouldn't even make it to sexual maturity.
You got it. Yep. Yeah.
So before we get to the within species pattern, because that's interesting that it's different from the across species pattern. So across species, smaller animals tend to live less long lives than large animals. There are outliers, as you mentioned. So tortoises famously have long lives. I believe naked mole rats live way longer than you would expect. When we look at those outlier species, do we know what the mechanisms at play there that make them outliers, and do they converge on anything, or are they all unique?
Yeah, it's a good question. I would say we don't. We can talk specifically about naked mole rat because there are a couple of interesting ideas there. Other than the naked mole rat, this hasn't really been studied in detail, and most of the studies that have been done done through, and this isn't meant to be critical, the use of the word biased, but through a biased lens, meaning people look at the hallmarks of aging, which gives us a handle for understanding aging, and they ask, if we look in very long-lived clams or other species, can we find evidence that these hallmarks of aging are decelerated or delayed? And so from that context, people have found evidence for things like, improved proteostasis. So proteins are either able to maintain their conformation better, or in response to some sort of stress that would normally cause proteins to misfold, they're better able to maintain, or they can degrade damaged proteins faster. So those things are true, but it's also true that people went looking for that. So you have to be a little bit careful not to draw causal arrows. So I think we certainly don't have a very good comprehensive understanding for why these species are able to apparently age at a much slower rate, certainly live much longer than smaller species, or in these outlier species, why they break that correlation. Naked mole rats are interesting, and there are a couple of people. So Shelley Buffenstein was kind of the first person to really make naked mole rats a very powerful model in the study of aging. So she's awesome. She used to go and collect these animals from Africa and bring them back into the laboratory. And so she's done a lot of work identifying, like I said, some of these mechanisms around proteostasis or anti-cancer mechanisms. And then Vera Gorbanova at Rochester is the other person who has identified a couple of interesting potential mechanisms. So one mechanism there that Vera found has to do with the type of hyaluronic acid that naked mole rats make. They make a very long chain hyaluronic acid, which Vera has proposed is involved in mediating some of the potent resistance of naked mole rat cells to cancer. So I don't know that they really understand the mechanism there, but they have been able to provide evidence that, at least in culture, this very long chain hyaluronic acid kind of prevents the naked mole rat cells from becoming cancerous and being able to develop some of the phenotypes of cancerous cells. So
What exactly is that molecule? Is it the same thing that you see in skincare products?
it's exactly the same thing. Yeah. So it's a natural metabolite. And this gets very rapidly to the edge of my understanding of hyaluronic acid, but it's my understanding that even humans make hyaluronic acid of various lengths. And but naked mole rats, for reasons that I don't think anybody knows, make a very, very long chain hyaluronic acid, which can impair. I think it. I get a little bit outside of my area of expertise, so I think it has to do with the ability of the cancer cells to metastasize, and I don't really understand why, but it's got something to do with the process of going from, it doesn't affect, I don't think, the oncogenic mutation, but it's the process of going to a metastatic cancer that it attenuates. Whatever the reason, the naked mole rats are very clearly highly resistant to cancer. So for a long time, people thought naked mole rats didn't get cancer at all. And then Shelley showed that in a few of their very, very old animals in their colony, they were able to detect tumors, but it's at a much, much lower rate. And Vera's model is that it has at least something to do with this hyaluronic acid isoform that they make. And Vera's actually done an interesting experiment where she took the naked mole rat gene for this long chain hyaluronic acid and put it into laboratory mice. And the effects are kind of modest, but it looks like there's a hint at least of a beneficial effect just from moving that one gene over into mice. So pretty interesting.
Interesting. Do you think it might have something to do with them being subterranean? And I'd imagine that environment is different in many ways, including air composition.
Yeah, absolutely. It's a good question. I don't know about the hyaluronic acid. Presumably, there's some reason why they evolved this very long chain form. But certainly other aspects of their physiology, like resistance to hypoxia, the naked mole rats are super resistant to hypoxia. So there's all sorts of physiological mechanisms that are going to be adapted for that subterranean environment. There's also the social aspect. So, naked mole rats have this social hierarchy where they have a queen, right? And then a bunch of, I'm not going to get this exactly right. Shelley's explained it to me, and this is, again, not my area of expertise, but they have one reproductive female in the colony at a time. And that's, again, a very different social structure compared to most mammals, right? In some ways, it kind of more resembles bees, again, from my superficial understanding, which also have some interesting aging properties, right? So is there a social, an evolutionary component to the social structure of this species that has influenced their aging phenotypes?
Maybe they're less stressed because they don't have to worry about competing for mates.
That could certainly be part of it. Yeah.
Yeah. That's actually, who knows, but there could be something to that.
Yeah.
I want to just ask you a vague question and give people a sense for the different sort of mental models that are out there around aging. So the question is simply, what is aging? Or to say it a slightly different way, what are the different ways that people out there generally think about it? And maybe an anchor point here is, one end of the spectrum could be it's a completely passive process. We're just accumulating damage over time, like our cars. And another side to this could be that it's a very tightly regulated, sort of pre-programmed thing.
Yeah.
How do people think about it?
Yeah. So I think that's a good framework, because I think there are different lenses. First of all, I think that word aging is somewhat problematic because people come into it with their own preconceived definitions, right? So just as an example, because I've been doing this for 20 plus years now, when I think of aging, I immediately gravitate towards biological mechanisms because that's what I've been studying, right? Most people don't think that when you think about aging. So I think it's useful to kind of put those frameworks around it. So fundamentally, I think what you asked is the question that often comes up, which is aging programmed? And by programmed, that word really means, is the biological aging process selected through evolution to happen? In other words, is it something where it is a program in our genes that is going to occur because it was selected for that reason? And I don't think there's much evidence in humans or most other animals for programmed aging in that context, that it was selected for. There are a few cases, like salmon, that you can point to where there clearly is this defined programmed aging process, or programmed senescence, I guess I should say. So I don't think there's much evidence for that. I tend to believe that aging is more a byproduct of an absence of selection, where once you have done your job of reproducing and passing your genes on to the next generation, there's really not a lot of reason for natural selection to prevent aging. And I think intuitively that makes sense, and I think that lines up pretty well with most of the data that's out there on the genes that affect longevity. They don't appear to have evolved because they regulate longevity. They appear to have evolved because they regulate growth and reproduction. And I think things like mTOR and these nutrient sensing pathways are a really good example for understanding that. So mTOR is one of the better characterized longevity proteins or genes. And what mTOR does is fundamentally it senses the environment and helps the cell or organism make the decision whether it should grow and reproduce, or if the environment's not favorable for growth and reproduction, shut down growth and become stress resistant, right? Mm-hmm. It turns out that one of the key features in the environment that are really important in that decision-making process is food, right? Yep. There's not much food around, really bad idea to have babies because you don't have anything to feed them. So mTOR primarily senses nutrients. When there's lots of food around, mTOR gets turned up, that accelerates growth and reproduction, also accelerates aging. When there's not very much food around, mTOR gets shut down, that slows growth and reproduction and promotes a stress-resistant state. The delayed aging we see with turning mTOR down is probably a byproduct of that enhanced stress resistance. And people have speculated that that's because, again, from an evolutionary perspective, it makes sense to survive long enough until the environment changes and becomes favorable and there's food around.
Yeah.
So I think that's a really good example where it's pretty likely that mTOR evolved to regulate growth and reproduction, and it's kind of an accident that it regulates aging, but it's really the growth and reproduction aspect that's important.
But you've got these little molecular machines like mTOR and other things. It looks like you've got a sort of a programmed aging program, but as you said, it's really just a byproduct of the fact that they're regulating growth and reproduction, and what's being selected is when an animal actually reproduces.
Right, exactly. And I think this is where a lot of the arguments over programmed aging kind of get off track, because people can be meaning the same thing, but to one person, programmed means, and this is what I believe it means from the evolutionary literature, that natural selection has acted for that reason.
Yeah.
It could still be a program. It's just that's not why that program was put in place. The program is really to control development and reproduction, not aging.
Yeah, that makes sense. When we think about, like things like mTOR and the connection with things like diet. So again, in evolutionary context, really, ultimately, what selection is always going to be selecting for, in some sense, is reproductive viability, getting you to a place where you can reproduce. Obviously, if you're a longer-lived species or it takes you longer to mature, you have to have add-on mechanisms that prevent you from dying before you get to that point. So that's where you would talk about things like anti-cancer mechanisms in a larger-bodied animal, say.
Right, exactly.
If you get to the point where you reproduce, at that point, selection can relax. And so what you're saying is that's why we see this connection where you have something like mTOR controlling growth and reproduction. And a byproduct of that is necessarily that's going to affect longevity in some way. But it's really there in order to cause the animal to grow when conditions are appropriate for survival and reproduction.
Yeah, exactly. And I mean, to tie this, so actually one thing to say on that is, Michael Rose did some very sort of important classic experiments a few decades ago now in fruit flies, where he was actually able to show that in these organisms where you can undergo many, many generations, you can actually select for increased lifespan by selecting for animals that reproduce later. In other words, if you take a large enough population of animals, and you only allow those individuals that reproduce later to pass their genes on to the next generation, you can, over many, many generations, actually select for longer lifespan, kind of for what the reasons you were saying is that if you're only allowing those individuals to pass their genes on, you have to kind of evolve mutations or mechanisms to allow for that later reproduction to make it that far. So I think that's a proof in principle of at least part of this idea that it is possible to evolve those kinds of longevity mechanisms. Obviously, I mean, he wasn't able to do those experiments to the point where you could get fruit flies living as long as mice, but who knows, if you could carry those out long enough, maybe you could actually start to see something similar to the magnitude of longevity effects we've seen. We can see if we look across nature. The other piece here that's interesting, though, is this will take us full circle back to the body size discussion. This within species body size relationship to longevity seems to be primarily controlled by these growth signaling pathways like mTOR, insulin, IGF-1, growth hormone. Those are the factors that are determining the within species longevity and anti-correlation with body size.
Can we dig into that a little bit more? So within species, larger bodied individuals, they have more growth. That's why they're bigger. So things like mTOR and these other pathways are presumably kicked up in those individuals. That is leading them to live shorter lives on average?
Yeah, at least. So that is very clear when the signaling is determined during development, right? So in other words, I kind of said that backwards. The body size is determined by the amount of signaling through primarily growth hormone, IGF-1, and mTOR during development. If those signals are high during development, you get large individuals. If it's low, you get small individuals. And you get large individuals are going to be aging faster and shorter lived. And so there were sort of classic experiments done by Cynthia Kenyon in the 1990s in worms, where she showed that if you made mutations in the insulin IGF-1 pathway in C. elegans, you could double lifespan. So this is the sort of classic DAF2 mutations. And then there was also work from Andy Bartkey and others in mice showing that if you had mouse mutants where they were deficient in growth hormone or IGF-1 signaling through development, they would live 60, 70% longer. So, and then you've got dogs where we have this body size relationship where big dogs age faster than small dogs. The largest single genetic determinant of body size in dogs is this IGF-1 growth hormone pathway. So it all lines up that in the laboratory animals, you can in a targeted way, make these mutations, cause bigger animals to become smaller, and they live longer. And then in dogs, we've got this sort of natural population, pseudo-natural, I guess humans sort of evolved them, but it wasn't with the goal of evolving longevity. You've got these natural populations where you have this big variation in lifespan based on body size, and it turns out the determinant of that is IGF-1 and growth hormone. So it all makes sense.
So these larger bodied individuals, they don't live as long, and they've got higher levels of these growth pathways building them during development. That's why they're bigger. What do they actually die of that gives them a shorter lifespan? Do they get cancer more?
I think that's a good question. I don't know if we really know the answer to that. It may also be somewhat species dependent. So, mice get a lot more cancers than even dogs do. Dogs get more cancers than people do. I think cancer is kind of the low hanging fruit there, but I'm hesitant to say it's all of the story. In fact, I'm pretty confident it probably isn't. The one, I think, interesting tweak on this idea. So if you look in dogs, this is where I think the best data exists. If you look in dogs, big dogs seem to age more rapidly than small dogs do in pretty much every tissue and organ that this has been looked at in terms of pathology and kidney. And, you look across the organs, they get more cancers, they get more arthritis. The one place where that seems to be different is the brain. So big dogs do not seem to have a higher incidence of dementia, just matched to chronological age. If anything, they might be a little bit protected, which is also kind of interesting because there's data, and it's not super clean, but there is data in mice that high levels of signaling through IGF-1 and insulin signaling in the brain may actually be protective against dementia. So it may be a case where these growth pathways are actually doing something different in the brain in terms of brain aging, or at least dementia, versus aging in the rest of the body. The other thing that's worth mentioning here is there was a study done by Walter Longo, which is very cool, where they went and studied this population of people who have a growth hormone mutation. They're called Laron dwarfs. And they looked at people who were kind of genetically matched in the same village. I don't remember what country this was in, somewhere in Central or South America. And they looked at longevity and cause of death in these people. So you have humans who have low levels of growth hormone. They're small. It turns out there's no difference in longevity. compared to the big people in the same population. The cause of death is completely different. So the Larone people were, if not completely, almost completely protected against cancer and heart disease, and I think diabetes as well. But they had much higher rates of death due to alcoholism and accidents. That's what it said in the paper. So I don't know exactly what that means, but I suspect part of that is in humans, it's a little bit more complicated than, say, in dogs or certainly in laboratory mice. There's this social component to height. And this is, if you look in the literature on this, it's kind of interesting because, many, many decades ago, there was this sort of myth that taller people were healthier and lived longer. And I think that was in part because of the social benefit that accrues to being taller. But now that we've been able to look in larger populations and different populations around the world, that actually doesn't seem to hold up. And in fact, there's a little bit of a cost in longevity to being taller. But the actual cost is probably bigger than that, but it's sort of offset by this social benefit that accrues to being taller.
Yeah, so there's a biological cost because if you're bigger, you've got more of these growth pathways that were turned up during development, and that's giving you the cost.
Right.
But you get the social benefit of being taller. In almost every culture I can think of, generally, if you're taller, all things being equal, you're going to have more social status.
Right.
So one way to, I guess, to think about the population you were just talking about is maybe alcoholism and accidental deaths were higher in the smaller individuals because they had lower social status on average. They were more stressed. They were coping with that somehow.
That's my guess. Yeah. Again, it's a guess, but it's pretty plausible.
Interesting. So you mentioned that I think you were being careful with your words. You were saying mTOR, IGF-1, these growth pathways, when they're turned up during development, that's when you create a bigger body, and that comes with a longevity cost. What if they're turned up after you become an adult? That's obviously something people do on purpose sometimes.
Yeah, I don't think we know the answer to that. So again, my speculation is that most of the effect is determined during development, but a small amount of the effect is determined post-developmentally. And the data to support that, certainly with rapamycin, we know you can start giving rapamycin to old mice and you still get a pretty significant longevity benefit.
And rapamycin blocks mTOR.
That's right, turns down mTOR, yeah. And there's one study from, I know Nir Barzilai was part of this study. There were other people, sorry guys, there were other people on that study as well, just don't remember who, where they gave, I think it was an anti-IGF-1 antibody to aged mice and saw a pretty small but statistically significant effect on lifespan. So I think you can get some benefits by turning down these pathways in middle age, but it's not of the same magnitude that you get from doing that during development and creating a small body-sized individual. I'm trying to think if I know of any studies where people have intentionally turned up these pathways in laboratory animals.
In adults.
In adults, yeah, post-developmentally. I'm not thinking of any. I know it's probably you're referring to humans. We do this all the time, right?
Yeah.
So it's interesting because, for many years, like I remember when I was a graduate student, the anti-aging docs giving growth hormone were all the rage, right? And those of us in doing this work in the laboratory were like, but that's the opposite of what we do in mice. So my take on that in humans is I haven't seen anything that makes me convinced that growth hormone therapy in older people has a major longevity cost to it. There might be a minor cost. Also, I haven't seen anything that makes me believe there's a longevity benefit to it. So my intuition is it's probably not moving the needle much either way. I think
so we don't know, but if an effect is there, it's not big enough that we've noticed yet.
Yeah, exactly. Exactly right. I think testosterone is kind of interesting because that's also, going to promote growth, at least in certain tissues, right? And I haven't seen convincing data one way or the other. I think certainly from a quality of life perspective, I've seen, certainly lots of anecdotal stories of people who saw a pretty significant decrease in testosterone in their 50s, 60s, and I'm talking men primarily. Women, hormone replacement therapy, to me, and maybe I'm going to get myself in trouble, but to me, the data is pretty clear that that's net beneficial for more people than it's harmful in
and
women.
What does that look like in women? What is the hormone replacement that they actually use?
Yeah, estrogen is the primary component of hormone replacement therapy. I think there are a whole variety
And
of different.
is that typically for postmenopausal
Yeah,
women?
exactly. That's
Yeah,
exactly
okay.
the context. Yeah, and I don't know how much of this you've followed, but there was a pretty famous study that kind of came to the conclusion that hormone replacement therapy in women increased risk of cancer. And so that's why for many years, many doctors stopped prescribing hormone replacement therapy for postmenopausal, perimenopausal women. That I think the pendulum has swung back the other direction. And again, I think that there's very, very strong evidence that for many women, hormone replacement therapy has a lot of quality of life benefits and also, helps maintain bone density, helps maintain lean mass, both of which are important going into, postmenopausal aging in women. So I think the and the cancer risk seems to have been, I don't want to say it's an artifact of the study, but it has to do with the type of hormone replacement therapy the women they were studying were taking and the way that the data was analyzed. So I think it was largely exaggerated is the consensus at this point.
Yeah, and I imagine there's a lot of things that have to be balanced here. So if you, for example, if you had, say, a slight increase in certain cancers with a hormone replacement therapy, but at the same time, you're less frail and less apt to fall over, and you're less likely to die by other means. You have to think about all these things.
I think that's exactly spot on. And I think, just sort of removing ourselves from that specific case, I think that sort of overall risk reward analysis often isn't done in our current sort of, medical structure. It's like there's very much this risk aversion that if there's any risk,
if the one marker is above the one level for the one negative outcome,
then we just don't do anything, instead of saying, even if there's a little bit of an uptick in risk for certain types of cancer, the additional benefits from body composition, may outweigh that. And so that analysis often isn't done for sure. So anyways, that was a little bit of a tangent, but all of this is to say that, again, I haven't seen anything to make me convinced that hormone replacement therapy, these growth promoting hormone replacements, for hormones that decline with age in many people, that has a significant risk of accelerating aging. And I think for many people, it's pretty clear that it has pretty substantial benefits for quality of life. This may also be a place where mice and to some extent dogs, because of just the way, so I think the biology of aging fundamentally is very highly conserved, but the phenotypic manifestation of that is going to be different in different species. And I think in humans, unlike in mice for sure, and in dogs are probably somewhere in the middle, the phenotypic manifestations in elderly people who make it into their 70s, 80s, and 90s, is loss of lean mass, risk of fractures, frailty. That has to do, right, with maintaining muscle function and maintaining bone structural integrity in joints. So these growth promoting pathways, while they might accelerate other phenotypic manifestations of aging, are protective for those things that in humans are proportionally probably more important than they are in mice. So these studies in mice may not reflect exactly what is going to impact quality of life in people.
Exactly. Like lab mice don't have to worry about falling down the stairs or anything.
And they do get fractures. Like there have been people who've done, micro CT analysis of skeletal structure in aged mice at time of death. You can see spinal fractures and things like that, but we don't quantify that in the laboratory because the mice are still moving around in their cage, right? So you have to go look for it, and it doesn't probably limit lifespan the way it does in a lot of people. I think this is another place where there's this, I don't know if it's a controversy. There are people who are very vocal on both sides of the dietary protein discussion, right? And I think that may be another example where, yes, in mice, protein restriction, at least sometimes, can extend lifespan very robustly. But again, mice are not really experiencing loss of bone density or loss of muscle mass to the point where it limits their lifespan. I'd be very hesitant to tell, a 50-year-old, 60-year-old person that they want to go on a very low protein diet, right? Because of the risk you might run from inducing sarcopenia or inducing, loss of bone density. So.
Yeah, I mean that's another area where it's important to remember that we get a lot of benefit from doing highly controlled experiments in artificial conditions with laboratory animals, but also we're not looking at naturalistic conditions.
That's right.
If you have mice on a low protein diet and they live longer, well, they're also not running away from predators and things like this
that's
that
right.
might require them to use their muscles more.
Yeah. No, I think that's exactly right. So I think just being aware of the strengths and limitations of the different models that we use is super important. And of course, the laboratory is a very well-controlled, it's not completely sterile, but certainly compared to the real world, it's a relatively sterile environment. They're not being exposed to all the pathogens that, humans are walking around in on a daily basis. And so all of those things kind of change the dynamic. And so what works really well in a controlled laboratory environment, may not have the same effect in the real world. And this is something I think about and try to point out a lot, which is that you might be able to take an intervention, right, that increases, let's say we had a drug that increases lifespan by 100% in the laboratory, doubles lifespan of a mouse. It might double lifespan in a person, but if it also makes the person, highly susceptible to something that's going to kill them, you only need one event to kill you to offset a 100% increase in lifespan, right? And in the real world, there's lots of things that could potentially kill you. So you just have to kind of be aware that what we do in the laboratory isn't necessary, even if the biology is conserved, isn't necessarily going to work the same way in the real world.
Yeah. So you've done a lot of work on the molecular biology of aging. I want to talk about some of that. I want to talk a little bit more first about diet. Mm-hmm. So you mentioned protein. In general, consuming more protein means more mTOR signaling, correct? Mm-hmm. So what are important things we have to keep in mind here with respect to protein composition? So can you start talking about how certain amino acids specifically affect mTOR compared to other amino acids and what we should start thinking about instead of just thinking about protein generally?
I can, and I will. I'm hesitant to really emphasize that too much because I'm not sure it really matters in the context of the human diet. So like leucine is typically talked about as the primary amino acid that mTOR senses through proteins called Sirtuins, and that's pretty well worked out. So we pretty much understand how mTOR is going to be most strongly activated by leucine as a specific amino acid, other branched chain amino acids as well. But having said that, mTOR is also activated less potently by other types of caloric intake. And so again, one way to think about it is mTOR acts in a network of proteins that includes AMP kinase, which is a sensor of energy status within the cell. Mm-hmm. It includes insulin and IGF-1 signaling, which are going to be affected by carbohydrates and fats. So while from a biochemical perspective, mTOR is most potently activated by protein and leucine in particular, from the network perspective, the entire network is influenced by other types of nutrients and other things in the environment, oxygen level, things like that. Yep. So I think it's a little bit, it's useful to understand from a biochemical perspective. Yep. It's maybe distracting from the more important message if people are really worried about trying to restrict, branched chain amino acids in their diet or
maybe you restrict
like
your branched chain amino acids, but if you're overeating other nutrients and you're doing other things that stimulate mTOR, it's not really going to matter.
Yeah, and the other thing I would say is mTOR is not on-off, right? And you want, like even with rapamycin, when we give rapamycin to mice, first of all, we're not giving them enough that we are really hammering mTOR down. And it has different effects on mTOR signaling in different tissues. I think optimally, right, you might, and this is speculation, but you might speculate that if we could do this optimally, we might want to turn mTOR down, transiently in the immune system and, more potently in, I don't know, I'm just going to throw out kidney and liver, but not turn it down at all in muscle, right?
Yeah.
We don't have tools to do that. So we're sort of, stuck using crude things like rapamycin, but even there, the tissue distribution is going to be different. The kinetics of mTOR inhibition and activation are going to be different. So the way I sort of think about this is protein in your diet is probably, all things being equal, going to have a larger impact on mTOR signaling, but overall caloric consumption from a health perspective is probably going to have a larger impact on health. In other words, I believe, and I think there's some data to support this, that a high protein diet in the context of a healthy diet at optimal caloric intake is very different than a high protein diet in the context of a shitty diet eating way too much, right?
Yes.
And I think most of the data that we have that suggests that high protein is associated with higher all-cause mortality and higher cancer is in the context of populations where most of the people were eating too much and eating a low-quality diet. So what I haven't seen yet is a really rock-solid good study of people eating a really high-quality diet and comparing high protein to low protein. I've seen a couple of studies that tried to do this. I don't think any of them are to the point where I would want to say definitively that the detrimental, sorry, let me say this again. The increased risk of cancer, for example, coming from high protein is completely gone in people eating a very high quality diet, but that's what the studies I've seen suggest. But I do believe that these are going to be very different. And the other thing I would say is, I think that all things being equal, it also seems pretty clear that a high protein diet makes it easier for people to maintain or build lean mass than a low protein diet. I'm not saying you can't build lean mass on a low protein diet. Some people have, but all things being equal across the general population, I think a higher protein diet, most people are going to be more successful at maintaining or building lean mass. Obviously, you need to actually do some exercise to contribute to that, but I think the data is pretty clear on that. Like, I don't think anybody would really argue with that statement.
You mentioned, I want to ask you more about the tissue specificity of some of these pathways and aging in general. So you mentioned, for example, in dogs, right? The older dogs, the bigger body dogs are aging faster.
Yeah.
But you mentioned this interesting exception to that, which is their brains don't seem to fit that pattern. In general, I would imagine that on average, our tissues are all aging at roughly the same rate. How true is that? Are there any tissues or organ systems in the body that age slower than others?
I think this is just starting to be figured out. So people are just starting to develop tools that at a molecular level can start to quantify rates of aging in different organs and tissues. And really, it's just within the last 6 months that we've started to see some of these papers come out. So from a functional perspective, for a long time, we've been able to measure different aspects of organ and tissue functions. So you do cognitive assessments for brain, you do vision tests for eyes, you can do that for heart, you can do it for kidney. And I think the observation there is that it's a very individual thing, right? That in some people, they can maintain cognitive function very well, but their hearts go early, right? And we haven't really had an understanding of that. So I think now these tools are starting to come online where you can at least measure some aspects of molecular markers of aging from blood, but get signals from different tissues and organs. So these would include things like, epigenetic signatures that you can detect in blood, but that are coming from cells from different organs and tissues. So I think we'll learn a lot more in the next few years. There was a really interesting study just came out from Tony Weiss-Corey's lab at Stanford, where they used a blood-based test. I think it was proteomics, so they're looking at proteins in this case, where they're able to match signatures of organ and tissue aging. And I don't remember the numbers off the top of my head, but the take-home message is that in most people, you can detect, or in many people, you can detect signatures of early organ aging. But again, it's very specific. In some people, it's the brain, and some people, it's the heart, and some people, it's the kidney. The cool thing there, if these tests pan out, is for an individual, you could go get a blood test, and maybe you find that most of your organs look great, but your kidney shows signs of accelerated aging. Is it really aging? That's a different question, but it's probably a sign of higher risk of kidney pathology compared to liver, brain, heart. And so you can then do some diagnostics to assess that more functionally, and then maybe do some personalized interventions to slow that down or maybe even reverse it. That, I think, is the real power of these tools. When we can look at organ-specific aging, we can start to personalize therapies to be preventative and proactive and catch these things before it reaches end-stage disease.
Mm-hmm. On the subject of diet, so I know that one of the more robust results out there in terms of increasing longevity is caloric restriction. What do we really know there, and what are maybe some of the things that people are being misled on?
Sure.
What exactly is the caloric restriction that leads to the longevity benefit? Does it matter what the exact macronutrient profile is?
I think that's still TBD. So first of all, I have to say everything that we know about caloric restriction and longevity, almost everything comes from laboratory studies in mice and rats. I mean, this has been done in other simpler animals, but let's just focus on mice and rats. The little bit we know in people suggests that caloric restriction certainly will lead to weight loss, and that will often improve some of the biomarkers that we have long associated with health, like blood pressure and things like that, lipid profiles. And there are some early indications that these molecular markers of biological age, like the epigenetic markers, are sometimes reduced by caloric restriction in people. Is caloric restriction a potent longevity intervention in people? No idea. And again, we can talk more about that. There are lots of reasons to think it may not be, at least across the population. So in laboratory rodents, what we know with certainty is that it is possible to achieve up to about 60% increase in average lifespan. So a big effect from about 60% caloric restriction below what's called ad libitum. In other words, letting the mice eat as much as they want to, or the rats. Unclear whether the macronutrient ratio matters. There have been, and this has been work that's been done since really the 1930s. So there's a large body of literature that is a little bit contradictory. I would say, again, it is at least the case that you can achieve these effects on lifespan more or less independent of which macronutrients you restrict. So people have done specific macro carbs, protein, fat, all three, two of the three. And in every case that I'm aware of, people have been able to extend lifespan. So I don't think we really know. It is the case that if you restrict sort of across the board, you get the most consistent benefits. Okay, so what are the things people have been misled about? One, caloric restriction always works. Seems like about a third of genetic backgrounds, caloric restriction does not extend lifespan at a given level of restriction. And in many cases, it actually shortens lifespan.
Oh, interesting.
So there's a strong genotype dependent component here. That should shock nobody. Everything is gene by environment, right? But unfortunately, this sort of sometimes gets represented as if caloric restriction always works. And no, there are many cases where it doesn't. And like I said, somewhere between a third and maybe even as high as a half, probably closer to a third of genetic backgrounds, a given caloric restriction paradigm will not extend lifespan. And in some fraction of those, it will shorten lifespan. Okay, so that's important to appreciate, especially if we're going to start recommending this to a genetically complicated population like humans or dogs. Another is this thing around timing. So, there's all this interest in intermittent fasting, time-restricted feeding. So I typically think of intermittent fasting as 24 hours or more, and time-restricted feeding as shrinking your window of eating within a 24-hour period. My take of the literature is that it is unconvincing that there is any longevity benefit from time-restricted feeding or intermittent fasting unless you also calorically restrict. And another way to say that is all of the benefits that people have claimed in terms of longevity and health span, really, from time-restricted feeding and intermittent fasting might be due to the fact that those animals were also calorically restricted.
So another way of saying that would be that the benefits people are seeing from daily intermittent fasting or time-restricted feeding don't obviously extend beyond the caloric restriction component of that. These are useful tools people have for restricting calories.
Yeah. So
that's really what's going on.
So I want to be careful not to immediately extrapolate to people because I'm talking about the mouse studies. Because remember, all of this idea that these are longevity interventions really comes from the animal studies because we don't know in humans, right? Nobody's done the clinical lifespan study, right? Nobody's going to. So I'm being careful in what I'm saying because I want people to understand everything you've heard about these diets as longevity diets comes from the studies in mice and rats.
Yeah.
And there, it is clear to me at least that there are at best minimal, and by minimal, I mean single digit lifespan effects compared to 60%. Probably not even that. at best minimal longevity benefits when you do these experiments in an isocaloric manner, where they are not calorically restricted. So the interpretation of that is all of the benefits come from the caloric restriction in laboratory animals.
I see.
So now what about in people? And I think I would align with what you said, which is that absolutely, these are really useful tools for many people to reduce their caloric intake or manage their caloric intake. Great, do it. But there's no evidence that I'm aware of that these are longevity interventions unless you are calorically restricted. And then I think we get into this sort of bigger question of, is caloric restriction a viable longevity strategy in people? And does it actually slow aging in people? I think there's reason to believe that it probably does impact the biology of aging in humans, similar to the impact that it has in rodents. But I also think in people there are all sorts of potential complications that go along with chronic caloric restriction, like the fact that we live in a complex environment, all of the social implications of caloric restriction. This was probably before your time, but when I was a graduate student, there was a very active group called the Caloric Restriction Society of people who were self-practicing caloric restriction. And, they had an active presence in the field. They would come to meetings. They had a very active discussion group online. And there were a lot of psychological complications that went along with practicing that diet in many of these individuals. And so I think that it's just really hard in people. And, there was a sort of high profile scientist named Roy Walford. I don't know if, again, probably before your time, but he's an interesting dude, actually. So he did a lot of the early mouse caloric restrictions, not early, we're talking 80s, 90s, caloric restriction studies with Rick Weindruck. And he wrote a book called The 120-Year Diet. And he actually was very active in kind of starting this caloric restriction society, and then he died in his, I think, late 70s. So, there are not and again, it hasn't been long enough to know for sure, there aren't very many people who can maintain that lifestyle.
Right.
And the sort of high profile cases didn't end well.
Yeah.
So I worry that is certainly, it's not even a worry. I am rock solid confident that is not going to be a strategy that you can use across the population. And I'm not sure that even if you could, that on average, it would be net beneficial for most people.
Mm-hmm. Aging is a concern for everyone. It's a problem that's been with us for all of human history. Nobody, for the most part, wants to die. Everyone wants to live as long as possible, or at least be as healthy as they can for as long as possible. The science has been progressing. Lots of exciting things are happening and have been happening for a number of years. You see a lot of chatter online from experts and from non-experts around aging and longevity. And people throw around terms like slowing down aging, or they might say stopping aging, or they might say reversing aging.
Yeah.
At a high level, how do you start to parse some of that stuff? Can we actually reverse aging that's already happened? Can we only slow it down, but we can't stop it?
I would say there's nothing theoretical that says we can't reverse aging.
Mm-hmm.
I think, again, this gets back to this issue of how do you define aging, right? And this is where I think, unfortunately, a lot of people are being very sloppy in their terminology. So most of the people who talk about reversing aging are talking about reversing one or a few aspects of aging, right? And so I think we've known for decades you can improve health, right? That shouldn't shock anybody. Is that reversing aging? I wouldn't say it is. Some people do, right? If you have gray hair that used to be black and you color your gray hair black, did you reverse aging? You reversed the phenotype of aging. That's really, I'm not saying that that's equivalent to reversing epigenetic age, but at some level it is. You've taken a phenotype of aging, which in this case is epigenetic changes, and you've reversed them. Did you reverse aging? Only if you believe that that's all of aging, right? So that's where I think the sloppy terminology comes in. What I would say is, that when we see interventions that improve health and improve function across multiple organs and tissues, then you can start to make a case that you have at least partially reversed biological aging. We have no data, and nobody has shown any evidence that they could take an old animal and make it young again. That's what I want to see.
What would that look like exactly in principle?
It would look like an old animal that looks young again, so physically looks young again,
Yep.
I would say, and also has restored the functionality of a young animal. So if you look at sort of obvious things like activity, movement, strength, look at tissue and organ level, you see a heart that's functioning like a young heart, a kidney that's functioning like a young kidney, immune system functioning like a young immune system. And then you look at the molecular level and you see all of these things reversed, right? That's a heavy lift. Like I'm not at all trying to say that that's going to be easy, but if you want to claim that you've reversed aging, then this is sort of my personal view. To me, claiming that you have made an animal age backwards is a pretty big claim to make. And so I would say it's really on the people making those claims to demonstrate. It's like it's not my job to prove that you haven't caused somebody to age in reverse. It's your job to prove that you have, right? So I think you really need to show that you can actually reverse at least the vast majority of changes that we see go along with age.
All the way down to the molecular level.
Absolutely. Yeah. Absolutely. And I think that's, like I said, theoretically possible. And there's reason to believe that, some of these new technologies like epigenetic reprogramming might get there, but nobody has done it yet. And so I think it's actually counterproductive to go around claiming that we've reversed aging. Because honestly, I mean, look, I don't know. I can only, like, everybody's frame is a little bit different. We all view the world through our own sort of lens based on our past experiences. But I gotta think, to the majority of people out there, when you've got scientists claiming that they've reversed aging, that the average person is going to look at that and it's just going to be like, that's crazy, right? I mean, maybe not. Maybe people actually believe it, but to me, that seems like that seems like science fiction. I think I would want to see somebody at least show in a laboratory animal that you can make an old animal young again.
What's an example in the literature from the last few years where a scientist has claimed to at least partially reverse aging, but you don't think they've demonstrated that?
I mean, there are lots. So you can go find, like, if you go look, if you just do a PubMed search for, reverse aging, there'll be plenty of titles. And let me say also, like, I think we're all guilty of, at least many of us in the longevity field are guilty of sloppy use of terminology. Like I've never used, I don't think, reverse aging that way, but I used to misuse the word health span all the time. So I'm willing to say that this is something that we all could do better at, right? So I'm not necessarily wanting to single individuals out. I think this is a pretty widespread problem. There are certainly some high-profile people who have kind of abused the terminology, but this is a widespread problem, and it's becoming wider spread because I think once a few people see individuals doing it and getting high-profile publication, it kind of feeds on itself. In the long run, maybe it's not going to matter. I mean, maybe what's going to happen is that phrase, reversing aging, is going to really come to mean in most people's minds slowing aging or targeting the biology of aging. They don't really believe that it's reversing aging. They just use it that way. What I would say, though, is, again, one of the problems I think with using that phrase reversing aging and trying to make it out like you've done something that's never been done before is just misleading. Because, like I said, we've known for decades, if you take a 65-year-old person who's overweight and sedentary and you put them on a healthy diet and you get them to exercise, functionally they're going to be better.
Yeah,
right?
And you would be able to measure a bunch of physiological markers that look younger.
That's right.
Yeah.
So this is not anything beyond what we've been able to do for decades.
Yeah.
And even like you look at rapamycin, and I certainly wouldn't claim that rapamycin reverses aging because it doesn't. It doesn't reverse aging in all tissues and organs. But you can take an old mouse, you start giving it rapamycin, its brain gets better, its heart gets better, its immune system gets better, its ovaries get better. So its oral cavity, you can reverse periodontal disease. So this is not anything beyond what we've been able to do for decades. And so it's kind of being presented as if it's this, shiny new object because the words are being used to mean something different than what they actually mean.
Yeah. The use of language is itself changing, and that confuses us. Say more about rapamycin. Let's just give people who don't already understand a brief overview. What is rapamycin, and how did it become that it was used in the aging world as opposed to its original use?
Right. So yeah, so rapamycin, super interesting drug. It was originally found from an expedition to Easter Island where they were looking for soil samples. And this is my understanding of that. The reason why they were looking for soil samples was because it had been observed that the people of Easter Island, which is also Rapa Nui, that's where the drug gets its name, rapamycin. The people of Easter Island, even though they went barefoot, didn't get tetanus, at least not at the rates that you would expect. So the expedition went there trying to look for microbes in the soil that might have production of molecules that would protect against tetanus. What they found instead was a bacterium in the soil that produced rapamycin. And so it was originally sort of developed. Well, it was observed that you could take rapamycin and put it on cells, and it had what's called anti-proliferative activity. So it basically slow the cell cycle of dividing cells. And so that got people excited for anti-cancer purposes. So some people started developing it for potential anti-cancer purposes, became a research tool that was used. That's actually how the protein mTOR was discovered, by treating cells with rapamycin and then looking for mutants that were no longer sensitive to rapamycin. But it got developed clinically as use as an immunosuppressant. And so that was how it was first FDA approved, was to prevent organ transplant rejection. And so we have a lot of, this was, 25, 30 years ago now that it was first approved. So we have a lot of data on rapamycin use in people in the context of organ transplant patients. And so that all happened, that was all happening in the clinical world. And then, as I mentioned, it was being used as a research tool to kind of figure out the biochemistry of mTOR and how the mTOR complex worked. And then this was around 2003, 2004. 4 different labs sort of all simultaneously got interested in mTOR as a regulator of longevity in yeast. That was work that I was doing, C. elegans and fruit flies. And I don't know about the others. Like, I don't think anybody was really talking to each other about this. It's like, this happens sometimes in science where you just all sort of converge on the same thing independently. And it all happened around the same time. And so in each of those 3 very evolutionarily divergent species, the 4 groups kind of independently found that you could genetically turn down mTOR and increase lifespan, slow aging. And so for me, when we first found that the mTOR mutant was long-lived, I had never heard of rapamycin. And so, of course, I went to the literature and found that, mTOR stands for mechanistic target of rapamycin. So the protein is actually named after the drug. We were genetically knocking it down, and there was this drug that could do the same thing pharmacologically. So it made sense to test whether the drug could also extend lifespan. So we actually did that experiment first in yeast, published that in 2006. And then I think that body of work, our work and the others in worms and flies, led to Dave Sharp proposing rapamycin to the interventions testing program as a molecule they would test. So the interventions testing program is a National Institute on Aging funded program to test drugs in mice for effects on lifespan. Still happening today. Anybody can propose a compound. You have to write a proposal, and then if it gets selected, it will be tested at 3 sites independently to see whether it affects lifespan in mice. So Dave Sharp proposed this. This must have been like 2005, right around 2005, that they started the experiment, and it extended lifespan in mice. And I think when it extended lifespan in mice, then everybody started paying attention. Before that, those of us who worked in invertebrates were excited about it, but I think, the larger community hadn't really yet caught on. But once we found that it worked in mammals the same way, then it really became, highly interesting and highly studied.
The clinical people started noticing.
Even then, I don't know if the clinical people did. It's kind of funny in this field, right? It's a little bit less today, but when I was a graduate student, you had a lot of people who thought that you couldn't learn anything about it. It's funny because I worked in yeast as a graduate student. So the worm and fly people were like, you can't learn anything about aging, studying aging in yeast. And then the mice people were like, you can't learn anything about aging, studying aging in worms and flies. And then the humans, the people who did clinical stuff, be like, you can't learn anything about aging unless you study aging in humans, right? So I think that mindset still persists, but it was more so even back in 2009. I'm not sure they started paying attention yet. But now we know. It's funny because when I first started in this field, there was a lot of debate about whether the biology of aging was conserved, right?
Yeah.
Now we know that's true.
Yeah.
We don't know how much of it's conserved, but a lot of it is. So certainly the broader field started paying attention when the mouse study was published. The other thing about that mouse study, though, that was really important and I think also sort of a paradigm shift was for sort of reasons that are funny. We can go into the story, but for reasons that are funny, they didn't start testing rapamycin until the mice were 20 months old, which is about the mouse equivalent of a 60-year-old person. Nobody thought that was going to work, but it worked. That was the first time a drug had been convincingly shown to have longevity benefits starting in middle age. Now we know that you can do that with other things as well. But at that point, I think for me, certainly, and I think for many people in the field, that really changed the way we thought about the plasticity of aging.
So at the time, you would have predicted that giving it earlier in life would have had this effect, but not that late.
Or it would have a tiny effect late in life. Yep.
But it was, how big was the effect?
So in the first study, it was about 14%, I think, which, at the time was pretty good for longevity interventions. And now that they've gone on and done dose responses, it's been pushed up to about 25, 30%.